Hello everyone. Can you guys hear me? Hello. Okay, great. So my name is Ashwant. I'm one of your PMRF TS for this course, Introduction to Cell Biology. Every week we'll be discussing the previous year's assignment questions and answers. And if you guys have any doubts or questions as well, we can try to discuss them during these sessions. So let me share my screen quickly. So can someone confirm if they can see the screen? Okay. So hello. Okay. Yeah. Yes. Tashifa Fatima, you have a question, or you cannot see my screen? Okay, everyone can see my screen, right? Okay, awesome. So the way I do it usually is we'll briefly go through the questions, and I'll give you guys a chance to answer and come up with an explanation. And if you guys are not able to answer, we'll discuss the problem in more detail. But if you guys are able to answer, we can discuss it very briefly and quickly move on. So with that, let's jump into it. Uh, yeah. So so the first question reads: Which of the following codon initiates the translation of mRNA? Hello. Okay, so the first question reads, which of the following codon initiates the translation of mRNA? So we have four options here: UUU, uracil, 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 UAG, AUG, or AUU. Do you guys know which codon initiates the translation? AUG for methionine. Yeah, oh, perfect. So if you don't mind, try to unmute your mic and speak because it's a little bit hard for me to shuffle between the chat and this. Let me see if I can. Just give me 1 second. I'll put it on another screen so that I'm able to see the chat also. Okay, so now I can see the chat as well. So let me do this. Yeah, so the correct answer is AUG. Uh, so essentially, we have these codon sequences, and we have basically these codes as to what they correspond for, right? And we have three stop codons in eukaryotes and one start codon, which is AUG for methionine, as I think Ajay has pointed out. Yeah, yeah. So UAG is stop codon, and triple U is so if you go through this list, triple U is basically for phenylalanine, and UAG is top codon, and AUU is isoleucine. So yeah, okay. Uh, so the next question reads: Which of the following is a property of intron in eukaryotic pre-mRNA? Introns are non-coding sequences that separate exons in monocistronic premature mRNAs, or introns are palindromic sequences that self-splice to form lariat structures. Introns are extra coding sequences that can be removed by an RNA protein complex, or introns can remain in mature mRNA due to alternate splicing pathways. So they're asking which of the following is a property of intron in a eukaryotic pre-mRNA. Okay, so Ajay is saying first option. Does anyone else have any opposing view or any other argument? So are introns coding non-coding sequences that separate exons in monocystronic pre-mRNAs? So we'll go through the options one by one. So introns are palindromic sequences that self-splice to form lariat structures. That's not true, right? Introns cannot self-splice. So intron splicing itself is a process in which there are enzymes that play roles. So and they're first of all they're not palindromic sequences. So that's not true. And introns are extra coding sequences that can be removed by RNA protein complexes. Is this true? Are they extra coding or non-coding? So they are non-coding. Okay, so a lot of you are agreeing that the first option is the correct one. Yeah. So essentially, introns are non-coding sequences that get spliced out of premature RNAs to sort of form this coding mRNA, right? So in this case, so if you see this phase zero intron sequence here, essentially these are symmetrical exons that are being spliced out to join these exons together and produce a mature mRNA. Okay. Which of the following is a property of? Oh, okay, so we're done with that question. So remdesivir, an antiviral medicine, is approved for authorized use in case of emergency to treat COVID-19 in many countries. Which step, which is described as depicted as the red numerical here, related to viral replication represented in the figure below, does remdesivir target? So, does it target DNA and DNA replication machinery? Or does it target RNA? Does it target proteins? Protein synthesis, RNA synthesis, DNA synthesis, translation, transcription. What does it target? Or protein mediated regulation of RNA. Do you guys have any idea? Okay, so Ajay is saying option D, which is 2, which is it targets the RNA machinery. Okay, yeah, so essentially that's what remdesivir is. It's an adenosine nucleoside triphosphatase analog. So it goes and interferes with the action of RNA-dependent RNA polymerase, which is very essential for viral replication and transcription translation, right? So yeah, so it severely decreases viral RNA production, and that's how it targets viral RNA. Okay. During transcription, dash strands of DNA, dash strands of dash is or are used as template. So is it one strand of DNA, two strands of DNA, one mRNA strand, or two mRNA strands? What happens during transcription? Yeah, so a lot of people are saying option one. Yeah, so during transcription, one strand of DNA is used as a template to produce what? So one strand of DNA is used as a template to produce what in a transcription? Yeah, RNA. And what happens during translation? How many strands of RNA are used? Yeah, so one strand of RNA is used to produce a protein, right? Okay. Yeah, so essentially, one strand of DNA is used to produce a template, is used as a template. Okay, so the next question reads, sorry. Yeah, so the next question reads, the formation of a lariat-like structure, which is the top left this not the ring-like structure that you see in this electron micrography image is characteristic of the process of, is it DNA replication, splicing, transcription, or protein synthesis? Okay, so splicing, people are saying it's splicing. Yeah, so essentially it's a characteristic typical structure that's observed during splicing. So if you look at the uh, biological process of splicing, introns form these lariat structures, and that's how they get spliced out, right? So this lariat structure that you see here is what you observe here. So that's basically the intron being spliced out. So lariat is basically a knot kind of structure. Yeah, yeah. Okay. So because there are four to the power three possible combinations of triplet codons, most amino acids are specified by more than one codon. This is the concept of what? Triple codon, equivalency, processivity, or degeneracy. So, do you guys understand the question? So if you go back to this codon chart, there are 64 possibilities here to code for amino acids, but we don't have so many amino acids, right? Generally, the biologically relevant amino acids are only around 21. So some of these codons code, some of these multiple codons code for the same amino acid. This is called what? So is it a triple codon? Is it equivalency, processivity, or degeneracy? Okay, so some people are saying degeneracy, some people are saying triple codon. Okay, so it's not triple codon. So I don't know what you understand by triple codon. Maybe you think three amino acids code for one, three codons code for one amino acid, which is not true. So sometimes there might be four codons that can code for one amino acid. In this case, there are three, but sometimes there can be two as well, and sometimes there can be just one. Uh, so usually, so if you look at tryptophan, it's only coded by just the one amino acid, right? So it's not triple codons. Uh, if that's what you understand by triple codon, if you're understanding saying there are three entities for to form a codon, then yeah, so that's a basic definition of codon. But the question they're asking here is most amino acids are specified by more than one codon, so it's called degeneracy. So this property is called degeneracy. So it's a fallback mechanism wherein you have multiple codons coding for the same amino acid. Okay. So the next question is, which of the following amino acids are an exception to Ramachandran plot? Is it proline and alanine, glycine and proline, or lysine and proline? Okay. So Ajay is saying glycine and proline. Any idea why? Why are glycine and proline accepted from Ramachandran plot? Okay, yeah. So essentially, they're non-chiral. They have aberrant structures compared to other amino acids. So they are the exception to the Ramachandran plot. So essentially, Ramachandran plot helps you predict. Okay, I'll not leak it. So, but this is essentially what a Ramachandran plot is for those of you who have not reached that point of the course yet. Yeah. So, which of the following protein structures can be predicted with the help of Ramachandran plot? So, we've seen this Ramachandran plot. Essentially, what it does is it will help you determine certain angles or conformations in which proteins can exist based on the angle of twist that the amino acid linkages can undergo. So, it will help you determine what structure. Is it the primary structure of the protein, secondary structure of the protein, tertiary structure of the protein, or quaternary structure of the protein? Okay, so Ajay is saying secondary. Yeah, so essentially, what the Ramachandran plot will help you determine is whether if a sequence of amino acids forms a beta sheet or an alpha helix. So both beta sheets and helices are secondary structures of protein. Okay. Okay, so the next question reads the omega angle of the peptide bond is acute, obtuse, right angle, or planar. Yeah, so a lot of people are saying planar. So yeah, so this this omega angle is planar where it can rotate to 180 degrees. Okay. The rotational span of phi and psi angles is what? 0 to 90, minus 90 to plus 90, minus 180 to plus 180, or 0 to 180. The rotational span of phi and psi. So if I look at phi and psi in the Ramachandran plot, what is the rotational span? Uh, is it 0 to 90, 0 to 180, minus 180 to plus 180, or what is the other option? 0 to 180. Third option. Okay, yeah. So minus 180 to plus 180 is the rotational span of pi and psi. Okay. So yeah, I think those were the questions for last year's week two assignments. So I feel like you guys found it fairly easy because we are at the beginning of the course. But do you guys have any questions or doubts or something that you're finding hard to follow? Yeah, go ahead. Someone has raised their hand. Okay. Uh, How are you guys finding the class? Are you guys mostly from biology background or engineering background? Okay. Okay, so biology background, you guys should find this fairly easy to at least in the first few weeks of the lectures. But if you guys have any doubts or any pressing questions, you can always bring them up. If you guys have uh, all questions already discussed in Saturday class. Yeah, so so since the course is huge, in case someone misses one of the classes, they have the flexibility to join Monday's class. So that's why we do it in on multiple days. But yeah, formula to calculate DNA strand after round. Um, Not sure what you're asking, but so if you're asking for the amount of the number of DNA strands after n rounds of, yeah, you can unmute. Feel free to talk.
Sir, Yashwant sir, there was
Yeah.
a question
Okay.
ki how many strands of DNA won't contain original strand after in non-graded question it was five. and then after 5 the answer was 30, correct there.
Correct, correct. So
And
this
then
is.
in actually quiz, first week's quiz they has asked ki how many stands means how many stands won't contain original strand
Mhm.
after 3 rounds.
Okay. So to calculate the number of strands after n rounds of division, so you will have 2 to the power n strands after n rounds of division, right?
So what I said to that Ankita madam is it is 2 raised to the power 3. So out of 8, only 2 original stand will be removed and it will be 6.
Correct. Yeah.
Huh. Okay, so madam said ki how she calculated, I don't know. She said ki it will make 12 stands, 6 stands will contain original DNA and
Okay.
that's why six, answer
Maybe
is 6. Okay.
maybe she got confused, but
No, sorry, because she got confused. Okay, I even got confused.
yeah, yeah. It's okay. It's okay. That's why.
Interestingly, what happened? There was another course of functional genomics
Mm-hmm.
in that madam PhD from Kanpur. Some madam was there, and she
Mm-hmm.
told me this is the formula. To raise to the power m means n or whatever the rounds are there.
Mm-hmm.
And then she calculated also with same question was there.
Mm-hmm.
Interestingly, I don't know how that happened, but
Yeah.
within a span of a minute, I got cleared. So the students attending the class, I got confused. How it is really 12 or 6? Because
Okay.
if after 50, I mean PCR or anything, they I think make 50, 60, 70 rounds. So that would only be possible to calculate on a paper. Like that. We
Yeah.
must do some
Yeah, yeah. Of course, you're correct. So the number of DNA strands after n
thanks for allowing me
rounds
to.
of no issues. Replication will always be equal to 2 to the power n. So so this is superscript. So 2 to the power n, right? So now if there are 3 rounds of replication, so that will be 2 to the power 3, 2 twos, 4 twos, 8. So 8 total DNA strands. And since this follows a semi-conservative mode of replication, there will be 2 parent strands, and the remaining 6 will be newly synthesized strands. Yeah. So for any of you guys that still have the doubt, is that clear?
Thank you.
Yeah. No problem. Do you guys have any other questions?
In a replication, wherever I found it is written semi-continuous and semi-conservative. But in course they had written discontinuous. Discontinuous means it cannot be semi-continuous.
So it's a contention between vernacular, right? So semi-continuous or discontinuous both mean to say that the leading strand gets replicated in a continuous fashion, but the lagging strand gets replicated in bits and pieces, right? So it's okay. So semi-continuous is also fine because ultimately whatever replication has happened, it's going to copy the entire. Mali Babu has a question, but yeah, so usually it's discontinuous or semi-continuous. You can say it's just choice of words. I think it's fine. So the important concept is it's not continuous. So that even though the leading strand DNA replication is continuous, the lagging strand produces these Okazaki fragments, and they need to be patched up by DNA ligase, right? So at the towards the end of the replication. So that's what discontinuous or semi-continuous are pointing towards. Is that fine? Okay. Yeah, Malli Babu, you have a question? You can unmute your mic and ask a question. Yeah.
Sir, I have a question.
Yeah, go ahead.
Sir, actually, we were asking about write the process about sporulation formation, but
Mm-hmm.
then in exam we got it like what's the process of from immature to mature state of sporulation. So what to write there, I didn't get.
I did not understand your question. So
Actually, we were taught only about what is sporulation.
okay.
Okay, then in exam we were asking about write the process of formation of sporulation
Mm-hmm.
from the immature state to mature state.
Okay. So you haven't been taught the formation of sporulation and development from an immature to mature state, is it? In the
Yeah,
core?
yeah, sir. Yeah, exactly.
Okay. So if you have not been taught, then you can raise an issue on the forums. So you know how to access the course forums? Hello.
Actually, sir, we have joined the college recently and we don't have that idea about it. And there is nothing like forum held in our college.
In your okay. So are you talking about the NPTEL course?
Sir, actually, our teacher taught us that it's a very useful course and you can join it there. So that we joined it. And here I got doubts, I'm asking you.
Okay. So in your college course, you've been so you want to understand what sporulation is.
Yeah, yeah, sir.
Okay. Okay. Sure. Let me pull up a diagram. Yeah. So let me share my screen. Okay. So essentially, sporulation comes along when you're talking about fungal replication and in some cases bacterial replication as well. Uh, Basically, there are several steps to this correlation. So there is vegetative growth in which fungus is growing as usual, but when it's presented with several environmental conditions, it might be harsh environmental conditions such as nutrient deprivation, temperature changes, so on and so forth. So there are these certain uh, genetic factors that can get triggered. So something along the lines of heat shock proteins that are very ubiquitously present. So when these get triggered, there is this triggered for formation of a spore. So it goes through various ways, various phases. So starting from early sporulation to late sporulation, and then ultimately the formation of mature spores. So in fungus, at least, this process takes place by the fungal organism generating this vegetative growth, and there is septa that forms. So basically, essentially, this cell wall will start forming and chopping up this vegetative growth into several distinct pieces, and these individual pieces will get sort of separated out, and these are basically the spores. So they have very thick walls and so on and so forth. So that's what sporulation is. Now, your question is, how do things proceed from early sporulation to late sporulation?
Yes, sir, yes.
Okay, so there are a lot of genetic uh, factors that play a role. There is a pathway for this transition from early sporulation to late sporulation. There are several genes that get triggered, activated, and govern this process. So I don't know how far you've been taught in your college and how much you want to go into detail, but you can take go to the website, go to the Google and search for early sporulation. Okay, let me pull that up also. Yeah. So there are several genetic factors that get triggered at various stages. So we know a fair bit of them, what these genes are and how they are getting activated. But there are essentially, I'm assuming what they've asked you to write is something along these lines, wherein you have vegetative growth under unfavorable conditions. There is which is followed by asymmetric cell division, in which there is a mother cell and which basically has contains a large portion of the cytosol, and there is also pre-spore, and it's an asymmetric cell division. So the distribution of nutrients and genetic material between mother cell and pre-spore is very different, and these two will start getting separated by a septum. Yeah, so essentially, once the septum is formed, so there is a pre-spore that has formed because of this asymmetric cell division, and the mother cell can then engulf this pre-spore. So that's stage three. And then there is a cell wall and cell membrane, very thick cell wall that forms. So at this point, there is no cell wall, but once the engulfment has happened, it will form a very thick cell wall within the mother cell through in a stage called cortex, and there will be spore coat as well. Depends on fungal species, which fungal species, and depending on which fungal species, the spore coat can vary in chemical composition, the size, the thickness, so on and so forth. And once the spore coat has formed, there is maturation of the spore. And remember, all this while, the fungus or in some cases, some bacteria are in this unfavorable condition. So the growth condition is not optimal. So the mother cell might slowly start to die. And if the mother cell dies, this spore is sort of protected from this harsh environment because it's engulfed within this very thick spore coat as well as cell wall. And if the mother cell dies or lyses, this breaks open, and that's what you see here, which is free endospore that has come out. At this stage, it is mature, and at this stage, it's early endospore. So early stages to late stages and mature endospore. So I'm assuming this is what they've asked you to write. I don't know how like what is being taught in your course. So yeah. So maybe you can just write something along the lines of during the early endosperm formation, there is vegetative growth of the mother cell under unfavorable conditions, which leads to asymmetric cell division. So you can also write about zearing, which pinches the mother cell and results in this asymmetric cell division. Again, there are several genetic factors that govern the formation of this zearing, and activation of these genetic factors is what promotes this zearing formation. But uh, essentially, because the zearing sort of pinches the mother cell, there is asymmetric cell division, pre-spore formulation, which is the early endospore formation, and there is engulfment, cell wall synthesis, spore coating, and then eventually maturation of the spore and cell lysis of the mother cell. Is that clear?
Yes, sir, it's clear.
Yeah, if they're asking about genetic factors that are that play a role in this, you'll have to read about it. So there are several genetic factors that get overexpressed when a fungal cell is growing in unfavorable conditions, fungal or bacterial. Yeah. Any other questions? Are you guys learning about the molecular biology of transcription, translation, and DNA replication as well? The enzymes involved, how it initiates, how it proceeds, how it ends in E. coli versus in eukaryotic cells and so on. Okay, if you guys have no, yeah, Ajay, go ahead. Do you have a question, Ajay? Okay, I guess not. But if you guys have no other questions, then how tRNA binds? Okay, how tRNA binds to the mRNA? Let me pull that up. Yeah. Yeah, so essentially, it all basically depends on the tRNA structure, which is the clover leaf structure that you see here. So tRNAs have something called the anticodon specific tRNA, how get attracted there. So you have to think about these biological processes as a mixture in a solution, right? So it's a random process. So it's not like a specific tRNA is getting attracted to the specific place. It's more based on the random chance of getting interacting with the particular mRNA. So wherever this translation is happening, which is in the ribosomes, there is a very huge concentration of these tRNAs, right? So and these tRNAs can have different anticodon sequences, and all of these interact with a particular sequence. And based on the binding affinity of a particular tRNA, it can stay or it can leave. If the binding affinity is low, meaning the anticodon sequence is not properly complementary to the codon sequence that's present in the mRNA, then the binding affinity will be low for the tRNA. So if another tRNA now comes with a stronger binding affinity, it will kick this tRNA out and bind instead. And whenever the perfect tRNA comes around with the perfect anticodon, perfect I say in quotes, but if you know what I mean, more or less perfect anticodon, then what happens is it will replace all the tRNA that has previously been sitting there with low binding affinity. So it's all basically in a mixture. So it's all happening in a mixture, and these things are just moving around, right? Like think of this like balls in a swimming pool. So like there are these balls that are moving around in a swimming pool, and you have so think of this like maybe there's a basket for these balls, right? So that's your mRNA. So so you have a bunch of balls of different sizes moving around in the swimming pool. Any ball can come and interact with this basket, but only the ball that will perfectly fit in the basket will sit in the basket. It will kick out any other ball that has been previously sitting in the basket. So yeah, so essentially that's how specific tRNA gets attracted there. It's a stochastic process. Tried each time. It will become too late. So first of all, this process happens in a matter of microseconds, the binding affinity, and it's not like all 20 tRNAs are getting tried every time, right? So there is so sometimes it might not come to 20 tRNAs. Sometimes it might just be that the very first tRNA that comes and interacts is perfectly binding to the codon, and these are in very high concentrations. Remember, so yeah, and this process happens very quickly. I'm trying to find the time scale. But yeah, so essentially this process happens within microseconds. So it's not and it's not like every time 20 tRNAs have to be tried out. How much time average RNA takes to translate assigned protein? Average. So it really depends on the size of the mRNA. If the mRNA is long, it will take a long time, but usually the translation happens in microseconds. Yeah, so I'm sure right now I'm pulling up a paper. So in prokaryotes, the translation rate is this. Let me write this down. Yeah, so in prokaryotes, 50 amino acids are translated per second. So requirements for formation of protein is spontaneous or it's like demand and supply phenomenon. Good question. So it really depends on the type of protein. So in a given cell, you have housekeeping genes, which basically code for essential proteins. So something along the lines of mitochondrial proteins that are very essential, or electron transport chain proteins, which are very essential for cell energy. Then these protein formation of these proteins continues to keep happening at a regular pace. But proteins can also be synthesized with a demand and supply model, right? So, and this really depends on the environment of the cell. So, if the cell is, let me give you an example. So, let's say cell is facing starvation. So, there are no nutrients around the cell. Now, the cell has a couple of options, but essentially, what will happen if there is no nutrient is autophagy mechanisms get triggered, right? So, then there is an upregulation of proteins that are related to autophagy, and then there is a downregulation of proteins that are not very essential. So, it's both. So, formation of protein is both constitutive depending on the nature of the protein. If it's a very essential protein for the cell, something like actin cytoskeleton protein that the cell requires always, then it's produced at a constant pace. So there is a constant turnover of the protein. But if it's a protein that's plays a very specific role in a very specific pathway, then yeah, so it can follow the demand and supply phenomenon as well. Okay. Okay, so if you guys have no other questions, then maybe we can end the session here today. I'll catch you guys next week with week 3 assignment questions. Okay. Uh, yeah, so if you guys have more doubts, please don't be afraid to ask them. So this session is for you to basically get a much more proper understanding of the subject matter at hand. If I'm not able to answer it immediately, I'll definitely get back to you next week. Uh, so, yeah. So, can you explain that? What's the concept of Ramachandran map? Okay. So, very briefly, Ramachandran map, the Ramachandran plot. basically calculates all the possible conformations in which amino acids can twist and a protein can therefore exist, right? So if you look at amino acid linkages, what happens is these bonds cannot completely twist forever. So they have a degree of freedom in their ability to twist because of steric hindrances, bond energy. So if they twist too much, there might be steric hindrances that might prevent them from twisting, or the bond energy might be too low that they cannot practically twist beyond a certain point. So essentially, what Ramachandran plot tells you is how much these linkages or bonds can twist with each other. So and what that will help you tell is so let's say this is an amino acid, this is an amino acid. So if you know how much this angle can twist, you know where one amino acid is with respect to the other amino acid. So as a shape. So if one amino acid is in one plane, is the other amino acid in the same plane, the plane above or the plane below, and where exactly it is, you would know. So if you know how much it can twist. So essentially, that's what the Ramachandran plot is plotting. How much uh, twist can happen within amino acid linkages and amino acids. And what it tells you is basically it gives you all the possible conformations an amino acid sequence can take. So essentially, there are, even though the twist can happen from -80 to +80, you might notice that there is nothing here. So that's because these twists are either not feasible or biologically not relevant. So what it basically gives you information on is how much of this twist can happen, and if the twist happens to a certain degree. So the phi angle is at some uh, -90, and the psi angle is at some 90. So what would that mean? So how is the protein how is the amino acid oriented to the next amino acid? uh, Too much confusion about angle. Ramachandran plot is on secondary protein, but angle is 3D. Okay, so secondary protein structure. uh, okay, so I see where the confusion is coming from. Okay, so angle is not essentially 3D. Angle is also 2D. So angle is basically the separation or the degree of separation between two lines, right? So it's a 2D measurement. It's not a 3D measurement. And so even though you see this thing in 3D, or you might try to see it in 3D, if you look at it from this perspective, it's just 2D. So angle cannot be measured in 3D. It can only be measured in 2D. And so beta, any analog we can see naturally like some tree branches. Okay, interesting question. I do not know. Let me think. Tree branches. Hmm. About angle. Okay. Uh, I'm not sure I completely understand, but so okay. So think of it this way, right? So you are trying to twist someone's arm. So you catch hold of someone and you're trying to twist his arm. There's only certain amount you can twist. You cannot twist beyond that, right? So if you keep twisting, there's only certain angle of twist that you can do. So if you take a hand, draw a line perpendicular to the hand, and draw a line. I won't try to draw, but if you take a hand, keep it straight, draw a line perpendicular to the hand, and draw a line, one more line that's perpendicular to the hand. So one in z axis and one in x axis, and you keep twisting. So you'll form an angle. So basically, that's the twist angle. Since it's angle between two amino alpha carbon carboxy atom. Yeah, so essentially it's an angle between two things. Angle cannot, or at least in biology, cannot be defined for three-dimensional space. Uh, It's always two things. The most simplest analogy that I can think is twisting someone's arm, right? Yeah. So if you take someone's arm, keep it straight, and you draw two lines perpendicular to the hand, one in the direction of the sky, the other one in the direction of the other one parallel to the ground. Now you have a 90-degree reference plane. Now if you twist the arm, there is an angle that's forming, right? So that angle is the degree of twist. Okay, so now where is psi, phi, and omega? Okay, so psi is essentially between the carbon atom of one amino acid and the alpha carbon atom of the other amino acid. And phi is essentially between the alpha carbon of one amino acid and the nitrogen atom of the same amino acid. And omega is essentially the nitrogen atom of one amino acid and the C minus one carbon of another amino acid. Could you explain protein folding? Okay, I'll have to really think about the hand analogy and try to see where psi, phi, and omega are in the hand analogy. I just came up with the hand analogy right now. Uh, yeah, I'll try to think about it and try to give you guys a better explanation in the next class next week. But yeah, I cannot think of where the psi, phi, and omega are in the hand analogy. I'll think about it and I'll get back to you next week. Could you please explain protein folding primary, secondary, tertiary, and quaternary? Okay, let me pull up uh, primary, secondary, tertiary, and structure of protein. Okay, let me paste this here only. Okay, so essentially, what the primary structure of protein is, I suppose the forearm could be phi and the bicep could be psi. Yeah, yeah, the forearm probably. Let me think about it and get back. But essentially, so the primary structure of protein is so. How is a protein formed? It's formed by amino acids linking with each other, right? So one amino acid, next amino acid, next amino acid, next amino acid. So if you take this sequence and write it down on a piece of paper, you know what amino acid follows what amino acid. So you know at position 1 there is some amino acid, at position 2 there is different amino acid, position 3 there is some other amino acid. So you know the sequence of the protein. So you know which amino acid comes first, which amino acid comes last. So that's the primary structure of the protein. So essentially, it's the polypeptide, it's the amino acid sequence with the polypeptide chains. So each amino acid is connected to one another, and that's essentially what the primary structure of the protein is. Now remember this. Initially, when proteins are being created, one amino acid is being added to the next amino acid, right? So that's when you will try to see at least very briefly this primary structure. But what happens is these amino acids are charged and they have hydrophobic they can have hydrophobic groups, they can have hydrophilic groups, so on and so forth. And they have a charge and they have isoelectric points. And this protein is existing, this amino acid sequence is existing in some solution, right? In our case, it's mostly water. So it's existing in a water-based solution. So it cannot exist like a chain. What will happen is, depending on whether if an amino acid is hydrophilic or hydrophobic, and depending on the charge of the amino acid, uh, so if an amino acid is hydrophobic, it doesn't want to interact with water. If it's hydrophilic, it wants to interact with water. So what will happen is, if an amino acid is hydrophobic, it will start to sort of rush inside and try to fold within the chain. So let's say if this amino acid is hydrophobic, it will start to come like this. And if these amino acids are hydrophilic, they'll try to surround this amino acid, right? So this amino acid is rushing inside. So that's essentially which results in the form, well, not technically true, but it results in. Okay, before we go there, so you see these bonds, right? So the covalent bonds through which these amino acids are linked. So as I was telling previously, so these bonds can twist and turn, and they can twist and turn to certain degrees, and certain degrees are allowed and certain degrees are not allowed. Depending on these twists and turns, they can form either alpha helices or beta pleated sheets. So these are the secondary structure of protein. So alpha helices and beta pleated sheets, essentially how these bonds are twisting and turning with each other. So if this amino acid bond twists, it will sort of come in the next plane, and if this twists again, it will come back. So it will form this spiral-like structure. And so on and so forth, right? So it will either form this spiral-like structure or it will form this beta pleated sheet. That's the secondary structure. Now, if we go back to the composition of the amino acid itself, they can be hydrophobic or hydrophilic or charged or not charged, several different configurations, right? All of this will result in uh, the folding of these amino acids in such a way that they minimize the energy, they minimize the entropy of the system, right? So essentially, all the hydrophobic amino acids want to not interact with water and get very deep into the amino acid core, and all the hydrophilic amino acid want to push out and be exposed to the water. So when this happens, so if you take it, I'm giving you an example. If you take this chain of amino acid and drop it in water, so immediately it will sort of go through the secondary structure and immediately form the tertiary structure, which is all the hydrophobic cores are inside, hydrophilic cores are outside, and so on, and hydrophilic layers are outside, amino acids are outside, and so on and so forth. So that's basically what the tertiary structure is. And this process, remember, also happens within the secondary structure. So this hydrophobic, hydrophilic repulsion attraction also happens in the secondary structure. So essentially, what this tertiary structure is made up of is the secondary structure, which is alpha helices and beta pleated sheets. And essentially, what the secondary structure is made up of is basically the primary structure, which is the amino acids sequence. Now, from amino acid sequence, we are going into alpha helices and beta pleated sheet based on the rotation of the bonds within the amino acids. And this rotation does not randomly happen, right? It happens because of several properties of the amino acid, whether it's hydrophobic, hydrophilic, charged, uncharged, so on and so forth. So, depending on these features, it will form these alpha helices and beta pleated sheets, which are secondary structures, and further interactions with the environment will force the protein or the formed alpha helices and beta pleated sheets. Now, one amino acid sequence may form two alpha helices. One alpha helix is hydrophobic, and the other alpha helix is not so hydrophobic. Now, this not so hydrophobic alpha helix wants to stay on top of the hydrophobic alpha helix. So, when these interact with each other, it will form this tertiary structure. Now, once a tertiary structure is formed, quaternary structure is essentially several of these tertiary structures coming together and linking with each other, either through hydrogen bond or metal ion bond, so on and so forth. So that's the quaternary structure of protein. I hope that's clear. But if it's not, please. Yeah. Okay. Great. Okay. So if you guys have no other questions, then we'll end here. I'll see you guys next week. I'll remember to explain you what the phi, psi, and omega angles are in the forearm analogy. Okay. Yeah. Thank you. Thank you for joining. Have a good night. Bye.